Hello and welcome to Nikolai's genetics lessons, and today's question is short fragments of RNA synthesized by primase during DNA replication are called, and here 5 answers to choose from. And first of all, let me explain how replication happens. Imagine that this is double-stranded DNA. Of course, double-stranded DNA would be twisted, so this is simplified version that suits my needs to explain how replication happens. And say bacterial DNA, bacterial chromosome would be circular. So this is how bacterial chromosome looks like, and those of eukaryotes are linear. So this is how human chromosome looks. And in bacteria, usually there is single origin of replication. So if bacteria would have a single point where 2 strands of the DNA would separate, eventually the whole chromosome would be replicated, and the process would end up on the opposite side, or in some bacteria this process goes in not 2 directions but in single direction, producing leading strand on both strands of the DNA. So in this case the whole process would end up at the same point where it started, but here is say position of the origin of replication. In different organisms we can find different sequences here, but what would be similar, that in most cases this would be a T rich sequence. Why? Because between adenine and thymine, between these 2 bases, there are only 2 hydrogen bonds, and between cytosine and guanine, triple bond. So of course in such origins of replication we can expect that this sequence to be AT rich. In prokaryotes like bacteria there is only single such origin of replication. In archaea we can find many such origins of replication along the chromosome, and in eukarya, in humans, we can find thousands of origins of replications, because our genome is 1,000 times bigger than genome of the bacteria. So in order to effectively replicate say chromosome, human chromosome has many origins of replication. Actually this is not to scale representation. Our 23 pairs of chromosomes in a single cell has about 100,000 of origins of replications. So every single chromosome in our organism has thousands of origins of replications. So now let's go over how the whole process works. So normally, if we would consider, for example, bacteria cell, bacteria chromosome, usually we can find it in not this form, but in a form that looks like this. So it represents negatively a supercoiled chromosome coiled on itself. And what I mean under the negatively supercoiled, I mean that for example, if this double-stranded DNA we consider it to be the best comparison would be with a spring, metal spring. So imagine that this is metal spring. So it twists in this way. What would happen to this metal spring if we would twist in the same direction? So we can expect that coils would become more dense, like what I'm showing right now, and also our chromosome would condense. So as you see, this would be more condensed form. But what would happen if we would proceed twisting it even more? Then this DNA would have so much strain that this would form a supercoiling. So would form such structures that would release tension and would create new such side chains. So for example, if we would twist 2 more times, we would create 2 more twist here. If we would twist even more, once again 2 more times, we can get second structure that also would have 2 twists here. So this twist would release tension. We cannot create tension for forever. So this would be something that we can see if we would do this experiment with a rope. But a rope wouldn't be very good example because rope doesn't have internal tension. So rope in this form would be relaxed. But DNA in this form would be coiled, and that means that it would have tension. So for example, the DNA in this form, in order for 2 strands to be parallel, we have to apply a lot of force to uncoil it. So it's not going to be in relaxed form. It's going to be under certain tension. And if we would twist or untwist our double-stranded DNA in the different direction, so in the opposite direction, and this representation of the double-stranded DNA when it is in the relaxed form. So this is not relaxed form. This is going to be relaxed form. But what would happen if we would start to untwist it? So before I show you example when we twist it in the same direction. But what would happen if we would start to untwist it in the different direction? It's never would become like in this picture, but what would happen actually the distance between coils would increase. First it would increase, and the length also would increase, but we also can do this to a certain degree, and with a next twist it would snap into the negatively supercoiled DNA. So once again making another coil here. So if we would proceed untwisted, we just would create more and more negative supercoiled DNA. So when we uncoil this coiled DNA, we create tension. This is the same, just consider this is not a rope, consider this is being a spring, metal spring. If we would start to twist it in the opposite direction, this would snap instantly and would create such structure like this, negative supercoil to release a tension. So normally in bacterial cell, single chromosome, circular chromosome, is found as negatively twist supercoiled DNA. Why? Because it's release tension and would add tension at these places, origin of replication, helping to separate these two strands of the DNA. And we also can find some bacteria in positively supercoiled form, and usually this is extremophils. So bacteria or archaea that found in hot springs, and no wonder, because this condensed form, even supercondensed form with positively supercoiled DNA, would protect DNA from unwinding better than relaxed form or negatively supercoiled DNA that actually helps to unwind DNA for excess when for excess of different machinery, cell machinery, for example, to make a copy of the certain fragment, which we call gene, or this helps in replication of the single chromosome. But in human genome or eukaryotes, we do not have negatively supercoiled chromosome or DNA because our chromosomes are linear. I think my explanation of positive and negatively supercoiled DNA would be one of the best on YouTube, because all the other examples that I saw myself used a rope as example, and rope doesn't work as good example, but a metal spring would be much better example how DNA can be positively or negatively supercoiled. But let's return to our problem. Another misconception. So today I want to talk a little bit about different misconceptions. Would be that, for example, you can see in textbooks replication fork, and here we have a primer, and here it is said that one strand of the DNA would replicate continuously from 5 to 3 prime direction, and another one would uh, would replicate discontinuously, making small fragments, which we call Okazaki fragments. But actually this is also not very true, meaning that here we see only one replication fork. But in order to see the whole picture, we have to talk about replication bubble. And now we would be able to see the whole picture. So imagine that somewhere here we had origin of replication, our two strands separated, and here we have primer. By the way, this is not DNA primer, this is RNA primer, would be added by primase. So basically this is going to be our answer, that short fragments of RNA synthesized by primase during DNA replication are called primers. So first primers are added, this is RNA primers, and say that here we have 5 prime end of this strand of the DNA and 3 prime end here, and then that means that other strand would have 5 prime end here and 3 prime end here. This also gives us information that this primer would have 3 prime end here and 5 prime end here. So any new strand of the DNA would grow continuously from 5' end to 3' end. So that means that here new DNA strand would grow continuously. No new primers are needed. And if we have here 3' end and 5' end here, so this primer would have 5' end here and 3' end here. So once again, new strand of the DNA would grow continuously from 5' end to 3' end. So would grow continuously here till this continuous new strand of the DNA grows, would hit another replication bubble in the case of the linear chromosome, or would end up its process on the opposite side of the circular DNA. But the rest of the same strand would grow discontinuously. So primates have to add new RNA primers here along this replication fork as it's going to be opened, and then would grow in backward fashion. So as you see, of course with the use of DNA synthesis, and as you see, the same strand of the DNA, new strand of the DNA, is grow continuously and discontinuously. So we cannot say that one strand of the DNA would grow continuously and another discontinuously. Every new strand of the DNA would grow continuously and discontinuously, just in relation to the origin of replication. And of course, as you see, our double-stranded DNA is twisted on itself. So in order to untwist here, there are such proteins like helicase that would unwind our double-stranded DNA, and helicase would create extra strain on the DNA. It would condense, and if this tension wouldn't released, would form positive coils. So in order to prevent this, there are other proteins that would help in this replication process that are called topoisomerase, which function would be to nick on one strand of the DNA, and this would cause rotation of double-stranded DNA around one strand. So when, for example, let me continue this DNA, when a nick would be created here, so as you understand, this would eventually lead to DNA to unwind, so to release this extra tension. By the way, also this fragments that we call Okazaki fragments. Why this is fragments? If you think that this would be continuous DNA here, actually not. Here and here and here and here we would have an X. So these fragments we would call Okazaki fragments, and in order to close this nicks, another enzyme would help, which called ligase, to close this nicks. Of course, even this presentation is oversimplification of the whole process. For example, I didn't mention that when two strands of the DNA would separate, also first of all. So let me use, for example, this picture. So let's imagine that this is strand of the DNA that's separated. In order to prevent two strands of the DNA to close again, single strand binding proteins would bind to single-stranded DNA. These proteins has high affinity to single-stranded DNA and prevent closing two strands of the separated DNA, or prevent such structures as hairpin structures when a single-stranded DNA would bind to itself. So on the overall, this is description of how replication happens. Of course, as I said, with certain degree of simplification. For example, I didn't mention anything about clamp protein and clamp sliding protein that helps DNA synthase to move along single-stranded DNA and extend new strand of the DNA. Without these two proteins, DNA synthase would produce only short fragments of the DNA. So these two proteins stabilize new strand of the DNA synthesis. And also I didn't mention anything about telomerase. As you know, eukaryotic chromosomes, which are linear at the ends, has telomeres that protect our chromosomes, and with each cycle of replication, the telomeres become shorter and shorter, but it doesn't mean that this protective caps on our chromosome is not extended with every also replication cycle. Enzyme which called telomerase would extend these telomeres, trying to restore the original length of the chromosome. But it's so made by nature that eventually these telomeres would shorten, and this is normal process, and of course this would lead to destruction of the chromosome, and if chromosome would be destroyed, the whole cell would die. But this is normal process because with a time different mutations may accumulate, which would cause may cause a cancer. So this is just mechanism how our organism protected from old chromosomes which accumulated a lot of different mutations. So let's return to our question. Short fragments of the RNA synthesized by primates during DNA replication are called primers. This is answer A. And this is all for today. Thank you for attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.